In this video, we will be reviewing glycolysis and all the steps along with it, in which it is the process that breaks glucose down to produce energy, or ATP. Glycolysis occurs in the cytoplasm of the cell and is extremely important for the synthesis of cellular metabolism. In this process of glycolysis, glucose is ultimately broken down into 2 pyruvates, 2 NADH pluses, 2 hydrogens, a net gain of 2 ATPs, and water.
To begin glycolysis, we start with 1 molecule of glucose. From this glucose, we can use the enzyme hexokinase to convert that glucose into glucose 6-phosphate. During this process, a carbon is attached to the 6th carbon of glucose, and ultimately 1 molecule of ATP is used up and lost. From glucose 6-phosphate, we can use the enzyme phosphohexose isomerase to convert that molecule into fructose 6-phosphate. From there, we can use the enzyme phosphofructose kinase 1 to convert the molecule into fructose 1,6-bisphosphoglycerate. During this process, another molecule of ATP is used up and lost. From fructose 1,6-bisphosphoglycerate, we can convert that molecule into 2 products with the enzyme aldolase. The 2 products are dihydroxyacetone phosphate and glycerolaldehyde 3-phosphate. In glycolysis, only glycerolaldehyde 3-phosphate proceeds through the reaction with the help of triose phosphate isomerase, and then it's converted into 1,3-bisphosphoglycerate with the help of the enzyme glycerolaldehyde 3-phosphate dehydrogenase. In this process, NAD plus and PI is converted into NADH and a hydrogen.
So in this next step, we have phosphoglycerate kinase, which is the enzyme. And this enzyme helps with the synthesis of 3-phosphoglycerate. Also in this process, we have the conversion of ADP to ATP. In this next step, we have phosphoglycerate mutase, which is the enzyme. And this enzyme is used in the synthesis of 2-phosphoglycerate. In this next step, we have enolase, which is the enzyme. And this enzyme is used in the synthesis of phosphoenol pyruvate. And in this process, water is produced as a byproduct. And this is the last step of glycolysis, in which pyruvate kinase is used as the enzyme that helps with the synthesis of pyruvate. And in this process, we have ADP to the conversion of ATP.
In conclusion, glycolysis is important as it is the initial pathway of cellular respiration. It converts glucose into 2 pyruvates, and during this process, a small amount of energy is produced. Pyruvate will be used in the future in the Krebs cycle.